in herbal medicine. In open access international book, Pharmacognosy Medicinal Plants, Dr. Selvakumari organized international national conferences and short-term courses. Also, she is an editorial board member and published over 28 review and research papers. With this brief introduction, I would like to invite Dr. Selvakumari Madam to give her insights on advances in natural product research through molecular techniques. Welcome, ma'am.
Thank you, ma'am. Thank you for your nice introduction. Is it audible, ma'am? Is it audible?
Yes, madam. It is audible.
Okay. I will share my slide first. Now my slide is visible.
Yes, madam.
Okay, thank you. So to start with, first of all, good afternoon to one and all who have gathered in this online platform for this faculty development program. And I would like to convey my thanks to the organizing committee members, to the principal madam, to the conveners, to the coordinators, and especially to Dr. Arifa madam for inviting me for the scientific session. And the scientific session entitled for my talk is Advances in Natural Product Research through Molecular Techniques, which is, I feel, more relevant to the theme of this faculty development program, that is Sustainable Innovation in Drug Delivery, a multidisciplinary perspective, precisely, if I want to say, from the pharmacognostical perspective. So going to the introduction part, there are remarkable contribution of plant products which is used as an API in global pharmaceutical market. So I just highlight a few examples of this API, plant molecule API, which we come across and we are well known. So morphine, which is obtained from opium. So this morphine molecule is available as intramuscular and subcutaneous injection. Morphine extended release tablets are available, and injections are available. So for as a narcotic analgesic. And the second example, what I highlight here is atropine from Atropa belladonna. So this is anticholinergic drug, which is used as a muscarinic antagonist, so which is being used in the treatment of organophosphorus poisoning. So this is available as tablet form and also in injection form, and also in transdermal patches are available in the pharmaceutical market. And the third example that I highlight here is vincristine injections, which is used as a chemotherapeutic agent against leukemia. And ergotamine and ergometrin injections are available, which is derived from ergot, which is a fungi, and caffeine, which is obtained from coffee arabica, from the coffee seeds, and Thea sinensis, tree leaf, which is used as a combinational drug in all the antihistaminic drugs to counteract the side effect. And so one more example I have given here, that is capsaicin, which is a pungent principle that is obtained from Capsicum annum, which is used as counter irritant, and it is available in the formulation of ointment. So these are the remarkable examples. And one more example I want to highlight here is digoxin. Even pediatric elixir is available for the pediatric use. So these are the remarkable example of API, that is the natural product API, that is available in global pharmaceutical market. So not only API, plant also contributes its own molecules as excipient in global pharmaceutical market. So this also we come across. Few examples I have given: acacia, which is obtained from Acacia arabica, trachagan. So these two are used in liquid formulating liquid dosage form. Starch, which is a very important binding agent, so for formulating tablet. So this is obtained from these different sources. And pectin, which is obtained from the citrus peels, so which is done by acid hydrolysis of pectin, that is being used in formulating targeted drug delivery system and also in formulating medical adhesives. And lecithin, which is obtained from the glycine max, which is a phosphatidylcholine, so which is being used as a very important excipient to formulate microspheres. And this beeswax, lanolin wax, so all these are used in formulating semi-solid dosage form. And lactose, which is used in dry powder inhalation for as in nebulizer. So these two or the examples that I have given, plants are contributing, not only plants, the natural resources, the microorganisms. So all these things are contributing its potent molecule in the global pharmaceutical industry. And I have highlighted the another example, that is orthomycin. So this is actually sesquiterpene lactone, which has been isolated from the Chinese traditional plant, that is sweet wormwood, Artemisia annua. And this molecule, because of this discovery in the 2015, Dr. Yu Tu received a Nobel Prize because it is a very potent molecule that is active against extremely drug resistant malarial organism, that is Plasmodium falciparum and Plasmodium vivax. And because of this discovery and its potent action, this World Health Organization has recommended this artemisinin as one of the combination therapy, which is called as ACT. That means that artemisinin based combinational therapy as a first line of defense against malarial infection. So the marketed name here is Ortiquick, in which we can see that artemisinin in the dose of 62.5 milligram with pipriquine with the dose of 675 milligram. So this for this discovery, Dr. Yu Tu received Nobel Prize in the year 2015, a decade before, from the traditional Chinese plant Artemisia. Another example, in the same year 2015, Microlabs has formulated a tablet uh, by using this Carica papaya dried leaf extract tablets. So they have formulated and they have gotten approval from the Indian regulatory agencies, that is from CDSCO, and they have prescribed, and now it is being prescribed by the doctors for the treatment of thrombocytopenia, that is the infection that is caused by dengue. So these are the two remarkable examples in which these two are the successful inventions that is being discovered from the natural resources, and it has been prescribed by the doctors, and it has been recommended by the WHO. So now, actually, we move on to the actual scientific content. So to start with, so if we do research in the plants, first thing is what the identification. So usually, what is the traditional approaches? We will go for the botanical identification, and we will go for, after identification, we will collect, we will authenticate, we will go for that is the botanical authentication. Then we will go for extraction process, then we will go for pharmacological activity. So then we will document the thesis, then we will submit, that's all, and we will get a degree. But when you go in the molecular aspect, that is in the advanced natural product research, so we have to involve the research in the molecular way. For example, if we go for the botanical identification, so nowadays mutation happens due to unfavorable conditions. The plants will morphologically will be changed, and what we authenticate botanically, it doesn't mean that it is 100% the same genus or same species. So in addition to the botanical authentication, we have to go for DNA authentication also. So that is the first step, that we introduce this advanced molecular research in pharmacognosy aspect. So this DNA authentication, which is also called as DNA barcoding. So what is this DNA barcoding? DNA barcoding means it is using the specific DNA sequence to identify plant species precisely in a robustic way. So that is what the DNA authentication studies. So this specific DNA sequence are called as DNA barcodes, in which is a very important genomics based powerful tool for taxonomical identification in a molecular aspect. And this DNA authentication includes two research areas. One is in the molecular aspect, that is the bench work, and second one, getting the result and the data, the DNA sequence from this molecular bench work, and we have to interpret those data in the computational aspect. So here I want to highlight, so what is the importance of this DNA barcodes, which has been recommended by the pharmacopoeias. So when we see this research article which has been published, so we can see there are some 1,133 medicinal plants that has been enlisted in different pharmacopoeias, in five basic pharmacopoeias, and so this is the details of those pharmacopoeias, in which it includes this many species of plants, genus of plants, and belongs to the corresponding family. So when you take Chinese pharmacopoeia, Indian pharmacopoeia, Japanese, Korean, and USP, so we can see, so this is the total number of species. When we calculate, it comes to 1133. This plants is available either in the product, in the form of as a API, or the excipient, or as a whole plant. So this is what the details about the plants that has been enlisted in different pharmacopoeias. And here when we see that, so this is the British pharmacopoeia. So this is the article being published, and this British pharmacopoeia has approved. So if you go for the that is the botanical authentication, in addition to that, you have to follow the DNA authentication of the plants for collection to isolate the molecules either as an API or an excipient. So when you isolate a molecule, before that, along with the botanical authentication, you have to go for the DNA authentication also. And they have recommended, you can see that British Pharmacopoeia has approved 6 annotated DNA barcodes. And you can see the plant names here, Anethum gravulans, that is ITS2 is the genetic marker. So you have to authenticate genetically by using this genetic marker ITS2 for this plant. And they have given another marker. So this is also ITS2, this is TNH, PSBA. So this is another genetic marker. So like that, they have given 6 annotated genes. So not only this botanical authentication is enough. So you have to go for, means they British Pharmacopoeial Commission have recommended what is the type of means what the marker you have to, genetic marker you have to use to authenticate genetically for the corresponding plants. And coming to the markers, so how we will select the markers? So in the previous slide, we have seen that, so they have given the markers. But when we go for research, so how we will select the markers? Because we will take a new plant to study the new type of phytometabolites. So for that, how we will choose the genetic marker to authenticate genetically? So either we will get the markers from the literature survey, or if it is not available in the literature survey, we can go for universal markers. So there are some universal markers out there. So RBCL, MATK. So we have seen PSBA TRNH, which was given in the British Pharmacopoeia, and this ITS. This is called as internal transcript spacer. So all these markers, when you take this RBCL marker, so this is chloroplast DNA. MATK marker, this is plastidal gene. This PSBA TRNH is the chloroplast gene, and ITS is the chromosome. So these are the different universal marker. That means that, so these markers will be available in all the plants will have these 4 markers in the respective genetic material. And so these markers, so these genes, so marker means this is called as genes. So these genes will not be translated. So because we know that DNA will have the coding and non-coding regions. So coding regions will be translated into mRNA. Then it will translate a message. But when you take this non-coding part, so it will keep its genetic information as a conserved. Means it won't share to the messenger RNA. So it will be conserved from generations to generation. So these are the genes that is present in the conserved region, so that it won't get mutated even in unfavorable conditions. So these genes won't get mutated, there won't be any alterations. So we can precisely identify, based on these markers, we can precisely identify the exact plant substances. So this is what the markers I told. And if we go for, so first step is we have to select the marker, and second step we have to identify the marker in the plant. So how we will identify? So this genetic marker, like when we use a standard compound at a sample solution, when we run a chromatogram either by HPLC or TLC or PC, so we will get a spot, we will calculate the ORF value, and we will match it. But here in genetic studies, it is not like that, because we can't able to run the marker and identifying the genetic sequence in our plant of interest. So it is not like that. So to locate this standard genes in our plant, so we have to use primers. So what are primers? Primers are short sequence of DNA. For example, so this is two standard. So for example, this blue and this blue is my RBCL. So what I will do first, I will unwind the supercoiled DNA. Then I will use this primers. This primers are the short sequence of DNA. You can see this. So these are the primers. So this is the two primers, that is forward and reverse primer for MATK, and RBCL. So these are the primers. So these are the primers. So this is called as forward and reverse primers. The two primers was given here. So what I will do, I will use this primers to locate. So we know that, so these primers, it will attach to the complementary base pairs. So if it is A is there, here definitely there should be T. If here is C is there, definitely there should be G. So like that, if my desired gene is available, if you want to check, I want to select the primer. So this primer is also available in the literatures. So what is the primers for which genetic markers which will be available in the literature survey. So for identifying genetic authentication, we have to know what is the, we have to means target what kind of marker gene, and what is the uh, respective primers for that. And so after finalizing this primers, how we will sequence it, how we will identify by using different molecular marker system techniques. So there are different marker system techniques are available. First generation, now we are moving into third generation molecular markers. And in first generation, um, so this is what the commonly used first generation molecular marker techniques, random amplified polymorphic DNA, and second generation AFLP, and third generation is SNP, that is single nucleotide polymorphism. So these are the common techniques that is being used for identifying the molecular markers by using the genetic markers and the primers. So this is what in the diagrammatic representation I given first generation, second generation, and third generation molecular markers. Now, so we know what is Murray marker gene. So either we can select from the previous literature survey, or by choosing any universal markers. And for locating the markers, that is the authentic genetic marker, so we have to use the primers. Then how we will do the research work in the lab. So first we have to isolate the whole genome from the plant material. So either the plant or animal or whatever the living organism, first we have to isolate the whole genome. So there are different methods are available. So what I have done in my lab is CTAB method. So this method will clean the nucleus, and it will liberate the genomes, whole genomes inside the nucleus. So I have used the CTAB method, and after taking that whole genome, so we have to run in 0.8% gel electrophoresis to visualize that whether the DNA is isolated. So whether we have achieved the desired DNA which has been extracted. So that, to check that, because RNA also comes, some impurity also comes. So to identify the purity of the isolated DNA, we have to run this 0.8% gel electrophoresis for visual confirmation of DNA. Then by using primers, so then we have to add this primers to the whole genome. Then we have to go for the PCR amplification. So this PCR amplification will locate, by using this primers, it will locate the marker gene which is available in the whole genome. And after that, we have to run again 1.2% agarose gel electrophoresis. So here we will get PCR amplified products. That is, the primers will form complex with the desired gene. What gene, for example, if it is ITS or RBCL, so it will form complex, and that will be visible in this PCR amplified products when running with 1.2% agarose gel electrophoresis. Then after that, we have to take that band that is being separated in electrophoresis, and we have to go for Sanger sequencing of PCR amplicon. So what the product we get by coupling with the primers and the marker gene. So we have to go for further. The next step is what? Sequencing method. So the normal available sequencing method, which is economically friendly, readily available, is Sanger sequencing. So we have to go for Sanger sequencing, in which, so like this, we will get our desired gene in the form of, this is called as DNA chromatograms. So like this we will get, so this color corresponds to guanine, green corresponds to adenine, and T corresponds to thymine. So the principle, it is depth actually. So we will add some dyes to the particular DNA nucleotides. So we will add some dyes, and the complex will be formed. So that is different area, but shortly I'm telling. So when we go for the Sanger sequencing method, so we will get a DNA chromatogram like this. So our desired DNA, and so this chromatogram is the output of the lab work. After doing all the steps, starting from the DNA isolation, visual confirmation by using 0.8% electrophoresis, then PCR amplification, then again checking the PCR amplified product, then we have to go for Sanger sequencing to get this DNA chromatograms. So after getting these results, we have to go for the computational work. So this is the bench work or the lab work, and this is the computational work. So here, by using these data, we have to go for editing of raw DNA sequence and multiple sequence alignment and phylogenetic tree construction. So what is this phylogenetic tree construction means? So we will get the result like this. The end product is like this, the circular or in this manner, linear manner. So this is what the barcodes. So this is what the barcodes means. This is the output of the genetic authentication. So how we will interpret, I will tell you in the next slide. For example, so this is called, so this is a linear one. So what I told, and this is what the result we got. So this is the phylogeny of Nymphaceae. So this is the genetic authentication. This is called as DNA barcodes. Okay, so this is called as DNA barcodes, and we can see different species, different. This is the genus, and these are the different species. So when we take this Nymphia alba is a species, so the same genus Nymphia, odorata, and alba is a different species. So when we go for the DNA authentication from this phylogenetic tree construction, so this called as phylogenetic tree, and this is will be obtained by this computational methods. So this shows that this alba is genetically somewhat related to Nymphia odorata, but it is not exactly same. It is genetically different. Okay, but when you take this Nymphia alba and Nymphia amazonum, so there is a vast difference. So it is genetically irrelevant. So like that we will interpret. If I am taking my genome and if it is correlating, if it is exactly coinciding with Nymphia alba, that shows that it is genetically authenticated. So for this genetically authentication, so not only the lab work we have to do, we have to go for the computational work also. For that, we have to be equipped with all this computational work, BioEdit, Genius Pro. So these are the different softwares, Mega X and POP and Master Base. So these are the softwares we have to we should get all the knowledge of these softwares for getting this phylogeny of this DNA barcodes. So this is how we will genetically authenticate. And so only botanical authentication, no, we can also correlate the genomes with the phylogenetics. So this is the linear form of DNA barcodes of Nymphia species. But so when you see this diagram, so this is a circular form. So this circular form also is the phylogenetic tree. So that is a linear construction. This is a circular construction. So that different options are available in the software. So we can see that. So this purple. So all this is having vitiferin A. So all these plant molecules are having vitiferin A, and you can see that. So the corresponding colors denotes the corresponding molecules. So we can correlate not only the plant identification, botanical authentication. So we can also correlate the phytomolecules with its respective genes that synthesize, that biosynthesize these kind of molecules. So this is the second advantage of doing this molecular advanced natural product research. Another two examples I have given in Ranunculale's order, so which is a circular form of phylogenetic tree construction. So you can see that molecular phylogeny correlated with phytochemical profiling. Even not only phytochemical profiling, we can correlate with plants or the families with ethnopharmacological data. We can correlate with plants with ethnobotanical data. So like this, we can able to correlate. Okay. So next one is here I want to highlight about the informatic tools, computational. So here I told about the computational tools. Uh, so here I want to give some inputs about these information tools or computational tools. So when you take this informatic tools, I told, so editing of raw DNA sequence, we will use BioEdit and Genius Pro, and multiple sequence alignment. We have to align the DNA what we get. So that DNA chromatogram, so that we have to align in a particular sequence. So by using this Mega X, and there is one search tool is available, that is blast algorithm. So I will just show it in, sorry, in For example, is my Google searches visible?
Yes, madam.
Okay, so I'm going to NCBI website. I'm just giving, it is National Library of Medicine, that is National Center for Biotechnology Information. It is an official website of the United States. So I'm just giving Calanco gastroenus, one plant name. See here we can see blast nucleotides. So we have to type this and we will get all the data. So all the data, and here we can see the nucleotides, and so these are the nucleotides which is being deposited. So this Calico is a plant. Calico gastrinus bonari is a plant. The nucleotides, you can see that ITS1, ITS2. So all these things which is being deposited. So what I will do, I will just match my genome sequence. So this is the sequence of this plant, which has been published by. So this authors, okay, and this is the Calanco ITS marker gene. Uh, so you can see that, so these are the sequenced genome by this authors. So I will take these sequence as a standard sequence, and in the software I will give the means I will give all the inputs of this particular sequence, and the raw sequence what I got from my research work, I will just match it. I'll just match it, and the software will say how much it is correlated, like that phylogenetic tree construction. So what is the distance, or exactly it is matching. So like that, it will match and it will tell you. So this is called as multiple sequence alignment, or for that we have to go for this blast search or run blast. So these are different tools. Just I'm giving the introduction, not the complete thing, because it will take months together to learn all these things. So blast algorithm. So one second. So by using this matching, I will construct this phylogenetic tree by using this Mega X, Pop, and Mr. Base softwares. So this is how I will get the phylogenetic tree construction with respect to the authentication purpose, or with respect to the phytochemical profiling, or with the ethnopharmacological details, or with the ethnobotanical details. And when we go for this, so these are the database which is available. So not only in NCBI. So I have shown only the NCBI database, how to show this, means how to retrieve the other deposited genome sequences. So there are other databases which is available. So I have given here, and so I will just show this. Okay, so this is what. ah. So in this, there are three genome was sequenced and deposited, and this is the species, the same plant with different gene, internal transcript spacer, so which we have deposited in NCBI, so for our plant. So Calanco, we can see that, so this is a Calanco gastronus bonari. So this is my student, Muthu Kumaran, and we have deposited uh, internal transcript spacer with different genome area. So we have deposited, and it was approved by the official website of United States government, and it was available in the website, so that it will be used as a standard markers for the global researchers to authenticate this plant. Um, one second, I'll take my. So this is one of the successful research we have done in advanced molecular research in the natural product. And so far we discussed about the genomic studies related to the authentication, the phytochemical profiling. So now we can also study the biosynthetic pathways, whether what is the role of the enzyme in the particular biosynthetic pathway. So for example, so the for example, we will take this ketosynthase, which is which is an enzyme that is involved in biosynthetic pathway of aromatic compounds. We know that anthraquinones was biosynthesized by poly beta keto intermediate pathway, and this ketosynthase is one of the important enzyme that is involved in synthesis of not only the anthraquinones, the flavonoid molecules, all the flavonoid molecules, chalcones, aromes. So what we have done, we have taken this enzyme producing gene, and we have referred the primers and the gene marker was available in the literature. We have taken those primers and we have isolated the DNA. So we have run the whole genome by 0.8% gel electrophoresis, and we have amplified with PCR primers. This primers are specifically, this will locate the ketosynthase gene producing gene. So this primers we have run with the in the PCR equipment, and finally, what the result we got is, this is the flower petals we have taken, because ketosynthase gene will produce flavonoid. Flavonoids are the colored molecules. So to identify that, we have taken the colored part of the plant. So that is the flower part, and we have taken the stamens also. So this is another method, by liquid nitrogen method. CTAB is one method, and liquid nitrogen is another method. So we have got the pellets of DNA, and we have run 0.8% gel electrophoresis. And so PCR amplicons we have again in 1.2% we have run this PCR amplicons, and you know that, so these are the different bands. So this is the DNA and this is the RNA. RNA will be in the lower part because it is having lower molecular weight, so it will resolute, means it will goes faster than the DNA. So DNA is higher molecular weight, so it will comes later. So unfortunately, so when we run the experiment, it is not available. So we couldn't able to find that PCR amplicons with that ketosynthase gene, because maybe due to the experimental error, or it may follow different pathway, not the poly beta ketosynthase pathway. It may follow shikmic acid pathway. So there may be a lot of reasons, but to highlight this, so these are the different molecular techniques that we can do it in advanced pharmacognostical research. And in addition to that, high throughput screening. So this is, we know that it is the robustic method to screen the lead molecules, 1,000 or 1 lakh molecules at a time, in silico methods, computational docking, and this is another method, CRISPR-Cas9 and gene expression profiling. So this is one of the technique, molecular biology technique. We can edit the gene so that the phytomolecule produce synthesizing gene. We can enhance that gene, we can engineer that gene, we can alter that gene, so that that gene will produce increased amount of phytometabolites, our desired compound. So that can be applicable, that can be achieved by this technique, that is CRISPR-Cas9 and gene expression profiling. And advanced analytical techniques, we come across HPLC. We know well about HPLC, GC, NMR spectroscopy, batch. As a pharmacognosist or a research person, we should know the method development. For example, if I'm doing analysis in HPLC, I should know what is the nature of the phytomolecules. What is the chemistry of the phytomolecules, whether it is highly polar or low polar. So what column, so what type of column. We know that atropine is a racemic mixture, which is positive and negative, which is DNL. So it is a racemic mixture. It can be resoluted by chiral columns. So we have to know that complete nature and chemistry of the molecules for the method development. So it is not that seeing the paper and just running the HPLC chromatogram, GC, NMR spectroscopy or other spectroscopic technique. First, we should know how to develop the method for the new molecules, and what are the different spectroscopical interpretation. So how to interpret. So everything we have to update with this advanced analytical techniques. And this I told, so integration of qualitative, like DNA barcoding and phytochemical screening in the previous slides, and genomic and transcriptomic studies. So transcriptomic means how the molecules, the gene that synthesize the primary metabolites, and it will be transcripted, like it translated to the secondary metabolite. So all these things we should know, all the techniques, all the molecular biological tool techniques to study these type of molecular works. So ultimately, molecular pharmacognosy is the only means booming area in which we can able to achieve a lot of our natural desired compound. And with aligning with artificial intelligence and machine learning in the terms of natural product research, so there are different AI analysis spectra tools are available. So I have given some only few examples, only 6 AI tools I have given. MS2 LDA. So this is the AI tool, that tool will retrieve the molecular fragmentation data, that is the metabolomics data, to identify the or to elucidate the structure. And Spec2 VEC is a natural language processing algorithm. So this is useful to assess the spectral similarities. So if we get different spectrums, we can able to understand what are the different spectral similarities, so that we can able to know it is a similar kind of structure of molecules. So like that we can able to understand. Then MS2 deep source is the MS fragmentation spectra tool. So it is a tool to analyze this MS fragmentation spectra that gives us similarities of the chemical structures which can be easily predicted. And MS2 query, so which is a machine learning tool that gives a similarity of the chemistry of the natural products. And INCHI is another tool, so which gives the data about the IUPAC approach phytometabolite structure. And SMART2, so this it uses GCMSMS database to analyze the metabolized compound. So here the challenging of the phytometabolites in the drug research is when we take a molecule for the experimental pharmacological work. So we don't know when we inject a molecule or we administered a molecule, phytomolecule, we don't know what kind of metabolites it is being converted. So what is the bioavailability? So how it is excreted? So what is the plasma level? So all these things, that metabolized product we couldn't able to analyze. So this software will give some idea about the metabolized compounds, what will be the metabolites that will be converted when it enters into your systemic circulation. And it is one of the very most versatile tool in metabolomics analysis for natural product drug discovery. So just now I told metabolomics, which is a very challenging one because we don't know what kind of metabolites, because it is actually a complex molecule. It will have chiral natures. So it is very complex. So it is very difficult to identify what kind of metabolites it is being converted. So for that, another experimental protocol I would like to suggest here is in molecular pharmacognostical research, we can use hepatic cell line. So here in this article, you can see that HEPA-RG cell line is being used, which is an in vitro tool for understanding drug metabolism and also toxicology in human. So we can use hepatic cell lines so that we can incubate our drug molecule in the buffer with the cell lines, and the cell lines will have the enzymes. It will metabolize those phytomolecules, and we can take that supernatant and we can go for the further spectroscopic techniques to elucidate what will be the metabolites that is being formed after incubation. So metabolomics also has to be concentrated. So far I have discussed about APA, but in uh, successful, sustainable innovations in drug delivery, excipients plays a very important role. So not only the API from the phytomolecules that acts as an API, even excipients plays a very important role in drug delivery. This is one of my very preliminary research that we have done. So we, as an excipient, in the part of the area of excipients, the role of phytomolecules which acts as an excipient. So when I was doing my PhD in the year 2014, when I have taken the TS of rhizome, I have observed the starch grains was dispersed in parenchymatous cells of this rhizome. So when I'm doing this microscopical studies, so what I thought is, okay, there are plenty of starch grains. So I have observed. Then what I thought is, okay, let me isolate this starch, and this may act as a novel starch when compared to other conventional starch, markedly available conventional starch. So I have tried in isolation part. The normal convention method I followed, and I got good result. So this is what the starch I have isolated from the rhizome, and so I have strained with iodine solution and observed in microscopy. So we can see that darker purple stain, and in polarized microscope I have examined it, and it shows some plus shaped and Y shaped marking on it, which gives the microscopic evaluation of that particular starch. And I have done the sim analysis. So from the sim analysis, I understood that the starch shape is different, not only a spherical, it is global, oval, so different shape, different size. So this is what I observed from the SIM, and I have done DSC, differential scanning calorimetry, and I have confirmed that. So this degrading temperature is 334.6 joules, which is similar to the potato starch, the degrading temperature. And I have taken that starch And in formulation of glimepiride tablets, I have used the novel starch instead of this potato starch. I have used this the starch which I have isolated with the help of Lifeline Pharma Industry. So we have formulated glimepiride tablet by direct compression method, and we have compared the DT time and this starch given only 2 seconds. So I have claimed that this starch is a novel diluent and also super acting disintegrant. Even for formulating dispersible tablet, we can use this kind of starch, and it was patented. I got an approval from the Indian Patent Office in the year 2022, I think. And coming to my conclusion, the advancement in pharmacognosy are not only improving the quality and efficacy of herbal medicines, but also it drives the innovations in drug discovery and drug development, offering promising avenues for future therapeutic interventions. My take home message: in every walk with nature, one receives far more than he seeks. Thank you. Any questions?
Thank you, ma'am, for your deeper insights on understanding biosynthesis of natural products, AI-powered spectral analysis, usage of hepatic cell lines in metabolomics, role of natural excipients such as novel isolated starch from ribosomes, which you have done in your PhD work. Wonderful, ma'am. We have got a lot of knowledge and we can use this for our future research work also. Thank you, ma'am.
And
Over to
all these
Arifa,
are
ma'am.
granted project, madam. So we have done through the a lot of financial gaining. So, and it is very difficult when we do as alone. So we can try in either way, by receiving grants or by group of research scholars.
Thank you, ma'am.
Any questions?
Over to Arifa, ma'am.
Thank you, madam. Thank you, madam, for the enriching session of the faculty development program today. On behalf of the organizing committee, I express our heartfelt gratitude to Dr. E. Selvakumari, madam, Associate Professor and Head in Charge, Department of Pharmacognosy, College of Pharmacy, Mother Teresa Postgraduate and Research Institute of Health Sciences, Puducherry, for sparing her valuable time and delivering an immensely informative and engaging session today on advances in natural product research through molecular techniques. Madam, your presentation has been truly enlightened us on the integration of molecular techniques with natural product research, which is offering a fresh perspective on drug discovery and development. The depth of knowledge you have shared, along with the real-time applications and future directions, has certainly added great value to this faculty development program. Once again, thank you. Thank you so much, ma'am, for gracing us with your expertise. We look forward to
Thank,
continued association and wish you many more successes in all your endeavors. Thank you, ma'am.
Thank you, ma'am. That is why I presented what the research I did. So not only collecting the data and presenting. This will be more fruitful, I thought, and I have
Yes,
consolidated
ma'am.
everything and I have presented. Thank you for giving the platform to the principal, to the management, and also
Thank you.
Arifa madam. Thank you very much. Shall I quit now?
Yeah, ma'am.
Yeah,
I
thank
have
you.
a small announcement regarding tomorrow's session. Dear participants, tomorrow's session will be at 3 o'clock, afternoon 3 o'clock, which is going to be held by, taken by Dr. T.G.K. Murthy, sir, Professor and Principal, Bapatla College of Pharmacy. Please join for the session tomorrow on time. Thank you. We are closing the session now.